Hello, and welcome to the Scope of Things podcast, a no-nonsense look at the promise and problems of clinical research based on a sweep of the latest news and emerging trends in the field, and what I think is worthy of your 30 or so minutes of time. I'm Deborah Borfitz, Senior Science Writer for Clinical Research News, which means I spend a lot of time with my ear to the ground on your behalf, and a lot of hours every week speaking to top experts from around the world. Please consider making this your trusted go-to channel for staying current on things that matter, whether they give us hope or cause for pause. In another 5 or so minutes, I'll be talking to medical oncologist Ravi Parikh, medical director of the Data and Technology Application Shared Resource at Winship Cancer Institute of Emory University, about a novel AI platform he helped develop to translate clinical trial results to real-world populations. But first, the latest news, including expectation setting for phase 2 cancer trials, key learnings about dementia from the Nun Study, links between cardiovascular disease and mild cognitive impairment, the use of aspirin to prevent cancer spread, a clinical trial map to improve study access, and a naturally occurring molecule that rivals Ozempic in its weight loss potential. A new study estimates that almost 20% of cancer patients in phase 2 clinical trials receive a treatment that is eventually proven effective enough to get approval by the U.S. Food and Drug Administration, which could have important implications for drug development and clinical trial recruitment. Phase 2 trials determine whether a new drug shows signs of efficacy, with all patients receiving doses intended for future routine use. The 400 phase 2 trials randomly sampled for inclusion had a total patient enrollment of just over 25,000 patient participants and tested 332 drugs. The FDA later approved 71 of these drug regimens, which 1 in 6 patients in phase 2 trials received. One important caveat is that only half to a tenth of patients meaningfully benefit from many FDA-approved cancer drugs. The findings could better set expectations for phase 2 trials, where most of the enrollees have advanced cancers that lack standard treatment options. Researchers at UT Health San Antonio recently analyzed more than 30 years' worth of data from the Nun Study, one of the most significant longitudinal aging and dementia studies of its kind. Key learnings are that some individuals show resilience to cognitive decline despite an Alzheimer's diagnosis, indicating unknown protective mechanisms are at work, and that early life linguistic ability and grammatical complexity correlate with a lower risk of cognitive impairment in later life. Findings were based on aging and dementia patterns of 678 nuns aged 75 and up from the School Sisters of Notre Dame, all of whom are now deceased. Research is now being performed on donated brain autopsy material and the extensive data collected during the nuns' lives. The European Union recently launched a major 5-year project dubbed Dorian Gray, which aims to uncover the links between cardiovascular disease and mild cognitive impairment, or MCI. Cardiovascular disease not only shares risk factors with cognitive impairment, but may also contribute to it, although a unifying theory remains elusive due to the lack of reliable biomarkers for early diagnosis and risk stratification in patients with the two co-occurring conditions. The goal here is to come up with a neurobiology-based hypothesis, untangle the connection, and reduce the burden of disease and prolong survival. Key parts of the project include use of data from over 300,000 individuals across 6 countries to develop an AI model for predicting MCI risk and predisposition, development of a digital coaching system to encourage healthy lifestyle changes, and a randomized clinical trial of an avatar-based exergaming solution. Scientists in the UK have discovered that aspirin could prevent some cancers from spreading by stimulating the immune system, specifically by decreasing a clotting factor linked to how aspirin works and releasing T cells from suppression. The mechanism, they say, will support ongoing clinical trials, notably the aspirin study that has been ongoing for the past decade, and could lead to the targeted use of aspirin to prevent the spread of susceptible types of cancer and to the development of more effective drugs to prevent cancer metastasis. It was previously observed that people with breast, bowel, and prostate cancers taking daily low-dose aspirin saw a reduction in its spread. The clinical trials now underway seek to determine how they can use it safely and effectively to prevent cancer spread, since aspirin can have serious side effects in some people. A clinical trial map has launched in the EU to provide patients and healthcare professionals with easy access to comprehensive, real-time information about studies being conducted in their area. The map can be found on the public website of the Clinical Trials Information System, and the first version is in English, with additional EU languages to be added in future releases. The European Medicines Agency, which is responsible for maintaining the website, recently hosted a public webinar to provide a live demonstration on how to use all the features, and a recording of the session is available for future reference. And finally, Stanford medicine researchers have identified a naturally occurring molecule that rivals Ozempic in weight loss potential, while sidestepping side effects such as nausea, constipation, and a loss of muscle mass. The newly discovered BRP molecule, made up of just 12 amino acids, acts through a separate but similar metabolic pathway as GLP-1, the hormone mimicked by Ozempic, and activates different neurons in the brain. A new company known as Merrifield Therapeutics has launched to move testing from mice and mini pigs to humans. Critically, the study used an AI algorithm dubbed the peptide predictor to weed through dozens of prohormones to pick out the relatively rare ones that divide in ways that create functional hormone peptides. As a reminder, you can find links in the show notes to the various studies, articles, and press releases to learn more about any of today's news items.
Are you enjoying the conversation? We'd love to hear from you. Please subscribe to the podcast and give us a rating. It helps other people find and join the conversation. If you've got speaker or topic ideas, we'd love to hear those too. You can send them in a podcast review.
It is now time for my chat with Dr. Ravi Parikh about a promising trial translator platform under development to help doctors like himself use real-world data to better know how to treat the patient sitting in front of them. Welcome to the show, Dr. Parikh.
Sure thing. Thank you for having me.
So happy you are here with me today. Thanks for taking the time. This new platform sounds sort of magical in that it addresses the age-old clinical quandary about whether a newly marketed agent or drug cocktail will work as well as it did for the average cancer patient in a landmark clinical trial. Was this largely a matter of unleashing artificial intelligence on loads of clinical trial and patient data and connecting the dots? Can you please sort of start us off here with a snapshot of how trial translator is able to accomplish this rather monumental translation task?
Sure. Well, let me first off by talking a little bit about a high-level description of the work and what data goes into the model. And so our intention with Trial Translator is to be heavily generalizable and pragmatic. We don't want to be using data that's only collected for small subsets of patients that can't really be generalized to the broader real-world cohort. So we base Trial Translator on routinely collected health data, meaning electronic health record information, genetic information, the types of data that are normally collected as part of anyone's routine care and are stored electronically. We just had high access to a data set that allowed us to collect that information across a large national cohort and mine it to really see which patients were responding to drugs as they were, quote unquote, supposed to in a clinical trial versus which were some populations that really had a generalizability gap when it came to clinical trials being applicable to them. In terms of what we did, it was largely a two-stage process. First, we applied a machine learning algorithm to disease-specific subsets of patients with either prostate, breast, colorectal, or lung cancer, and use the machine learning model to predict their risk of mortality. And then second, we emulated a clinical trial within those risk subsets that we termed high, intermediate, and low risk groups. And we tried to replicate or emulate a clinical trial within each of those subsets to see, well, in which populations did a clinical trial generalize well versus when it didn't? So it was really a two-stage process of first applying a machine learning algorithm And then second, trying to emulate a clinical trial that had already been done.
Gotcha. So let's get into some of the results. I mean, in this study that's recently been published, focused on the long breast, prostate, colorectal cancers. Tell us a little bit before we get to details. Give some context in terms of, I don't know, what's the big need here, and what you're trying to address. What are you trying to provide that will be of use to clinicians in these particular areas? Yeah.
So, I'm an oncologist, and when I get the results of a big phase 3 trial that's presented at one of our national meetings or coming out in a really prominent journal, the immediate question I always ask myself is, that sounds great, but is the patient who's sitting in front of me going to respond like the patients did on the trial? And that's an understandable question because of two things. First, we design clinical trial eligibility criteria, often in ways that exclude patients who we normally see in the real world, either patients who may be sicker or who may have certain comorbidities that are routinely excluded from trials. But even if for people who are theoretically eligible, we know that there's some systematic barriers behind patients participating in a trial versus those that don't. For example, clinical trials often require extra visits or more transportation. And so I think that the question of how clinical trial results generalized to real world populations is something that is just trying to answer a question that I and my co-authors ask all the time in a clinic. That's largely the motivation behind the work. I think what enabled us to sort of answer the question now is the availability of one, large national level real world data sets, like I was speaking about before. And then two, the advent of machine learning algorithms that could synthesize large swaths of healthcare data and design algorithms based on large amounts of covariate to sort of identify signal within the noise in ways that our traditional regression models were just not really able to do.
What are some of the big takeaways that we might want to share with listeners from this study? I mean, what did you learn that you can put immediately into practice?
Sure. So, I think the top level results of the study were, one, kind of writ large. Real world patients tend to enjoy less benefit of a novel therapy than patients in clinical trials do. What we found was that there was about a 6-month survival decrease associated with a novel therapy in the real world compared to in a clinical trial. And so those 6 months, is actually a pretty substantial amount in an advanced cancer population. So that's sort of providing some evidence that real world patients are different. But I think the more interesting results was that when we used machine learning to phenotype these patients, we found that some patients that we termed low risk populations, groups that generally were a little healthier, they tended to respond to a novel agent pretty similarly to how patients respond in a clinical trial. But for patients who were high risk, patients who generally had higher comorbidities or were sicker or were identified by the machine learning algorithm as being higher risk, those patients tended to enjoy much less, if not no benefit to a novel therapy, indicating that clinical trials really just generalized quite poorly to them, even among people who were theoretically eligible for the trial. So, one big takeaway would be, if I was using this in a clinical setting, I might be able to counsel a patient that was identified by the algorithm as high risk a little differently than I would a patient who was low risk and likely to enjoy a benefit of the treatment. I might be a little more cautious with a high risk patient. It just might not respond as well as we think. And maybe we ought to be thinking very carefully about adding on a drug that has likely a high side effect profile potentially when it's unlikely to offer potential benefit.
Yeah, and temper expectations, I would imagine, too, especially if they're being advertised on TV as some new life-saving whatever.
Absolutely. And I mean, I think all of our high risk populations, unfortunately, are really prone to being targets of messaging like that. This is a drug that could, save you, where other types of standard treatments can't. And of course, that's what we want for our patients. But we just have to be realistic about expected treatment benefits. And oftentimes, clinical trials don't give us that information, whereas hopefully something like Trial Translator could.
Right. But I believe it's also pointing to opportunities for new clinical research that needs to get done specifically for this high-risk population group. Is that correct?
That's right. That's exactly right.
Okay.
And I think that the other big takeaway for that's more relevant for perhaps some in your audience that are designing clinical trials is that it's these high risk populations that maybe we ought to be thinking differently about clinical trial design in terms of the types of therapies that we use or the types of management strategies that we use, because we can identify pretty proactively with pretty good accuracy whether these groups are likely to respond similarly versus differently compared to lower risk groups. And so, thinking about different trials for these people, I think is ought to be a big focus.
Is there some resistance to doing that for, I don't know, economic reasons or just old habits die hard kind of thing?
Yeah, I mean, I'll say that when I design clinical trials myself, I always have this issue between trying to use our eligibility criteria to create sort of quote unquote clean cohorts of individuals that are likely to be somewhat homogeneous and respond similarly to a given drug, versus not wanting to cut down a sample so much that it's going to reduce my sample size and power for a trial. So that's a sort of an age-old tension. But I think what we're trying to expose here is that a lot of those seemingly benign eligibility decisions that we make, if we exclude this person or if we make this decision about how we run a trial, we might exclude some people, but they're still likely to respond the same way. Well, that doesn't actually hold from what our study says. We can actually show that specific groups of real world patients that don't respond as well. And the reason they might not respond as well is either because we don't have enough evidence about them, or because they're not just generally the type of patients that doctors and other health system administrators tend to enroll in clinical trials. And I think one of the top line messages is that we ought to be trying to get these higher risk patients enrolled in trials much more often, either by designing specific trials for them or by including them in our eligibility criteria, and kind of going against some of that age-old sort of guidance that we get around cleaner cohorts, because they represent a big evidence gap for real-world oncologists.
Yeah. And I believe last we talked, you were speaking about the flexibility that's sort of built into the platform for answering sort of translational type questions. Can you talk a little bit about the flexibilities and the ways that Trial Translator can be used in a research context that might not be obvious at first glance?
Sure. Yeah, I mean, so in the paper we kind of emulated. We developed Trial Translator based on 4 of the most common advanced cancers in the U.S. and tried to show the results in 11 emulated clinical trials. That being said, there's no reason why the platform itself, which essentially, as I mentioned, consists of a machine learning risk stratification and then a trial emulation, there's no reason why that couldn't be applied to broader cancers. I think all it takes is a sufficient amount of real world data, and then a solid treatment question where we can sort of have enough data to emulate the type of clinical trial that's out there. So for a clinical trial that was recently done, we might not have all the real world data that we need right after it's been done to be able to do this sort of stratification. But for a disease that where a clinical trial was published a year or 2 ago and where there's sufficient real world utilization, we could sort of, and we have been trying to adjust the platform to incorporate different diseases and different types of data sets. And so that's why we called it less of an algorithm and more of a platform to be able to ingest this type of data to make real world insights.
Yeah, and you had mentioned also about resuscitating drugs that had previously failed in trials. I think there's going to be some interest in that piece of it. Could you just speak a little bit to that? And are there any that are being investigated yet or being talked about being investigated?
Yeah, so this is a really interesting angle of the Trial Translator work that we are starting to pursue in advanced bladder cancer, but is also very amenable to being repurposed for other trials. So, when we have a negative phase 3 trial, for example, our general tendency is to scrap that kind of treatment or that kind of management strategy because it's not better than some sort of standard of care. But within a negative phase 3 trial, there are subsets of patients that we know might actually respond to a drug that is deemed quote unquote negative or ineffective. And so, unfortunately, traditional clinical trials aren't normally designed to identify those subsets of patients. And so those drugs, it takes years to identify those subsets of patients, or maybe they aren't identified at all. And so a platform like Trial Translator can actually try to emulate or replicate a negative clinical trial and use combined clinical and genomic data to identify subsets of patients who might be more likely to respond to a given drug that was ultimately deemed ineffective. Now, that's not to say that doctors should be using that drug, but I think it informs how we might design future clinical trials that target some of those subpopulations that are most likely to respond, so that we can get more effective treatments to market, whereas we might not otherwise have done so.
Right. Wow, that's a great use of this tool. Yeah, toggling back to like the clinic use, as a regulatory grade tool, how far off is that? And what will it take to get there?
Yeah, I think I kind of think about a tool like this as akin to a digital twin, a computational representation of a patient. Because essentially what we have right now is a set of code for large populations of patients that consists of code for high, intermediate, and low risk patients that we know respond differently to drugs or standard of care agents. And so we can apply those digital twins to future studies, I think, as potential external or historical control arms to allow a more robust comparison in single arm phase 2 studies, for example. And that what it might do is allow a higher level of evidence in those phase 2 studies than is normally done with an uncontrolled study. So that's sort of something we've been really excited about, is the concept of sort of applying the phenotypes to more prospective settings that could be used in regulatory grade decisions. I think for a platform like Trial Translator to be used writ large, it would take something like that, use in a prospective use case, to be kind of garnered some sort of regulatory decision. But I think this is the kind of debate that we're struggling with a lot of AI-based tools, is what level of prospective evidence is necessary. That's sort of just my hypothesis of what would be needed.
Well, that sounds like a perfect segue to my last question for you here today, and that's your immediate next steps. I mean, for you and your team, I mean, where do you personally take Trial Translator from here over, let's just say, the next year to two?
Sure. Yeah. Well, I think, there's so much insight in real world data that we're just scratching the surface of with something like Trial Translator. So I'll give you an example. One of those is the negative clinical trials resuscitation. That is a keen area of interest. Another area of interest is, oftentimes in when it comes to trying to use real world data, we focus on endpoints that are a little easier to measure, things like survival or recurrence of a cancer. But there are other outcomes that matter to patients that are available in real world data. We just don't make the effort to extract them. Those are things like toxicities or adverse events. And so one interest of mine has been to show that real world data sources can not only emulate survival outcomes, but they can emulate adverse event outcomes, and in fact, potentially point towards hitherto undescribed toxicities from drugs that are evident in the real world that just might not be evident from a clinical trial or might not be reported by clinical trial patients. And I think that would add a huge unexplored dimension to something like Trial Translator, would be to try to predict individuals who may incur side effects versus those who don't, because that would allow us to counsel real world patients a little bit more, a little bit more readily than we currently do. Normally, I find it really difficult to know who's going to have a side effect and who won't. And so, we might not be utilizing all the preventative strategies that we could. And so hopefully something like this in the next year, if we're able to get high quality adverse event data, could shed some light into that.
Well, innovation born out of frustration.
Yes.
It cannot hurt that you're an MD who sees the evidence gaps that need filling firsthand. On behalf of patients everywhere, particularly the unlucky ones with cancer, thanks for putting your mind to this important work, Dr. Parikh, and thanks for taking the time to be a guest on today's show.
Thank you very much for having me.
And as always, a big thank you to everyone out there for listening in. If you're not subscribed to this podcast yet, please consider going to Apple Podcasts and doing so right now so you don't miss your monthly dose of news and perspectives you'll be hard-pressed to find anywhere else. And if you're up for it, I'd also be so very grateful if you'd leave a rating and review while you're there. For more straight talk on studies involving humans, visit clinicalresearchnewsonline.com. And if you are a clinical research professional, we hope also to see you at our next SCOPE conference, where we make things happen. Bye for now.